A probable cause of pregnancy sickness has been found, and it may lead to a treatment. Sickness in pregnancy, or hyperemesis gravidarum, is common and is thought to affect 7 out of 10 women at some time in their pregnancy. But until recently, very little has been known about why it happens. New research by our team has identified sensitivity to a hormone made in abundance by the developing pregnancy, GDF-14, as a contributor to the risk of pregnancy sickness. This condition can affect pregnant women's quality of life, even in so-called mild cases. Between 1% and 3% of women suffer from a severe form of pregnancy sickness when nausea and vomiting are so severe that they lose weight or become dehydrated, or both. In one study, this condition was the most common reason that women were admitted to hospital in the first 3 months of pregnancy. It has been associated with worse pregnancy outcomes, and its effect lasts beyond the end of pregnancy, with some women reporting psychological distress and being reluctant to conceive again. The fact that it develops in early pregnancy and invariably resolves when pregnancy ends strongly suggests that the cause of the sickness comes from the developing pregnancy. But the detail on how and why it happens has remained elusive. This dearth of understanding makes the development of treatments difficult and arguably contributes to the considerable stigma associated with this condition. What is GDF-15? GDF-15 is a hormone that suppresses food intake in mice by acting, probably exclusively, on a small group of cells at the base of the brain, which are also known to induce nausea and vomiting. As such, GDF-15 has been under investigation as an obesity therapy. Early trials confirm it suppresses appetite in people, but it also causes nausea and vomiting. It has long been known that it is abundant in human placenta and is present at very high concentrations in the blood of healthy pregnant women. These factors make it a plausible cause, but a detailed understanding of if GDF-15 affects the severity of sickness in pregnancy has been lacking. We used a variety of methods to study how GDF-15 increases the risk of pregnancy sickness. We measured GDF-15 in the blood of pregnant women attending hospital due to sickness and those attending hospital for other reasons. Now, what could be a roadmap to treatment? The conclusion of these studies is clear. Predisposition to higher levels of GDF-15 when not pregnant reduces the risk of pregnancy sickness. At first glance, this is rather perplexing, because how can having higher levels of a hormone that makes you sick protect against pregnancy sickness? In fact, several hormone systems exhibit a phenomenon resembling memory, where the sensitivity to a hormone is influenced by previous exposure to that hormone. This seemed like the most plausible explanation for our results. Supporting this theory, mice with persistently high levels of GDF-15 in their bloodstream were relatively unresponsive to an acute surge in GDF-15 levels. Findings conducted by the conversations suggest that lower levels of GDF-15 before pregnancy result in women being hypersensitive to the large amounts of GDF-15 being released from the developing pregnancy. This poses two obvious approaches to treatment of this condition: desensitizing women to GDF-15 by increasing its levels before pregnancy, or blocking its action during pregnancy. The current challenge now is to develop and test strategies to achieve these aims that are safe and acceptable to women at risk from this debilitating condition.